In this video, we will continue our discussion about the relationship between hGTSE-1 and p53 relationship. So in the previous video, we have discussed that hGTSE-1 will somehow direct p53 to prevent the cells from apoptosis whenever the cells are under stress through this ET and through another artificially introduced DNA damage process. So here we will only consider this DNA damage through ET. So in the first figure, what we have done is we have decided to use this silencing RNA uh, that can knock down this hGTSE-1 after treatment of cells for 40 hours with this kind of RNA. So in this experiment, the objective was to check whether this knockdown of hGTSE will affect the quantity of this p53 within the cells or not. And when they did it with the control silencing RNA that does not have the silencer for this hGTSE, in that case we can see the presence of hGTSE-1. And when the same experiment, when they treat the cells with silencing hGTSE, they can see that for 40 hours they can see that the hGTSE is almost completely knocked down. And when they check these cells and when they check these two cells for the cell cycle analysis, they can see that in the control case, the sub-G1 level that they considered it as the apoptotic signature or signal for the apoptotic condition of the cells. They can see that there are less apoptotic cells compared to the condition where this hGTSE is knocked down. So from this they can made a point that hGTSE somehow directs p53 to prevent this apoptosis, as can be seen in the first case or the control case. And this is the image analysis of the same this we can see that apoptotic cells in the control is apoptotic nuclei in the control is less compared to the case where this hGTSE-1 is knocked down. And when they did the quantitative analysis of these images from the confocal microscopy, they can see that in the case of control they considered this as a reference, and for the case where hGTSE is knocked down, there is an increase in the apoptotic nuclei. However, one interesting thing that they observe is that in the case of knockdown of p53 using silencing p53 RNA, they can see the reduction in the apoptotic nuclei even less than the compared case. And when they knock down both the cases, and this percentage of apoptotic nuclei is comparable to the case where they have knocked down both p53 and hGTSE. So from this they made a hypothesis that it is p53 or the presence of p53 that results in the apoptotic of the cells, and hGTSE is somehow reduce or control the production of p53. In order to test this hypothesis, they did another experiment, is that they use different concentration of hGTSE to check the concentration of p53, whether hGTSE will affect p53 or not. And the two of this concentration is shown here. In one case there is almost nil, no hGTSE. In this they can see the p53 presence, and when they treat the cells with, when they overexpress hGTSE, they can see the reduction in the p53 level. This is for clone 1, and this is for clone 2 also. Similar case they have observed. Clone 7, and when they use a specific kind of reporter to check, or luciferase reporter to check p53 presence, and when they did this analysis, they can see that in the empty vector where there is no hGTSE-1 overexpression plasmid is there, they can see the p53 signals are, or luciferase signals are high, and when hGTSE is overexpressed, they can see the reduction in the luciferase signal that directly relates to the p53 levels within the cells. So the hGTSE will somehow regulate the levels of p53, and the p53 amount of p53 will regulate the apoptosis of the cells. So the second hypothesis they need to check with this the next experiments. In this, what they did is they introduced this hGTSE and check the p53 levels. Along with that, they also check the hGTC level in this case. Let me repeat this experiment. What they did is that they did the same experiment as it is done above, and that is when they use the control, there is reduction in the amount of p50. This experiment they did in the, we can say, without any overexpression of p53, or without external overexpression of p53. It is just endogenous p53. They did the experiment with endogenous p53. So now I think this experiment will be clear. So when they use the control vector that does not have the plasmid to silence hGTSE, they can see the reduction in the amount of p53. And when they use the silencing RNA for hGTSE, they can see the increase in the amount of p53. And same thing they quantified with the luciferase signal for 53. They can see that high luciferase signals where hGTSE is silenced. However, the one interesting thing that they know that even though without stress condition the amount of p53 is their presence, it will not trigger apoptosis. So one of the hypotheses is that the phosphorylation of p53 that will trigger the apoptosis of the cells. So in non-stress condition, the p53 is not phosphorylated, could not be phosphorylated. So they check that hypothesis in the next experiment. In this case, with this in the no ET treatment, when they did the experiment with control, that is without knockdown of hGTSE, they can see hGTSE levels are high and p53 levels are low, and this is the antibody for phosphorylated p53. And this is the another protein that is produced when the cells are in the, when p53 triggers apoptosis, that are also low. And these are the control experiment. And in no ET condition, there is no DNA damage, when they did the experiment with silencing hGTSE, they can see the increase in amount of p53 and decrease in the amount of hGTSE. However, no phosphorylated p53 is expressed. So this this substantiate their hypothesis that p53 is not activated or not phosphorylated, thus it does not trigger the apoptosis. It has to be phosphorylated at serine 15 position, and there is no or very low production of this signature protein that is produced when the p53 triggers apoptosis. It is also low. In the case of ET, when they use the controlled vector, we can see the presence of hGTSE, and p53 levels are also reduced, or we can say p53 levels consider this as a reference or control. So these are the p53 levels that are expressed in the case of this empty vector or control vector, and they can see the phosphorylated p53. So in the case of the cells are under stress, or when the DNA damage is there, then the p53 is phosphorylated and then triggers apoptosis. That is that can be observed by this signature protein that is produced when the p53 triggers apoptosis, that is Bugs protein. And when they treat the cells with silencing RNA of hGTSE and uh, artificially introduced DNA damage or stress condition through this ET, they can see that reduction in the amount of hGTSE because of the silencing knockdown of hGTSE because of the silencing hGTSE RNA, and the phosphorylate the p53 levels are increased compared to the previous case. And also the phosphorylated p53 levels are further increased compared to the control case, and the production of Bugs protein is also increased compared to the control case. So in this case, since the hGTSE is knocked down, there is no upper limit control over the p53. So p53 expressed much more compared to its control case, where hGTSE is controlling the overexpression of p53. So and this thing is further quantified in the case with the help of luciferase signals. In the no ET condition, no stress condition, they can see the luciferase signals increased when the cells are treated with silencing hGTSE. However, they are far below when the cells are under stress and treated with and this hGTSE is knocked down, because at this condition there is no upper control over p53 expression by hGTSE. This is how there is a relationship between hGTSE and p53 uh, to allow expressing this p53 in accordance with the stress that is observed within the cells. So I hope you have understood the basic idea about this experiment and what are the hypotheses and how they use the experiment to test their hypothesis. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.